Hey folks, we continue here by talking about how we begin to process actual things. So once the signal gets to our occipital lobe, the striate cortex of our occipital lobe, how are we going to start differentiating one object from another and identifying objects out in the world based on their basic features? And this might seem like an easy task, but if you've ever thought about how difficult it's been to design self-driving cars or robots to take in visual signals from the world and differentiate maybe a bicycle from another car, you quickly realize how many problems actually have to be solved to be able to process that visual information and identify things in the world. So one problem is simply determining where one object begins and another ends. So how can I tell where the edge of a letter is? How can I tell where the edge of a picture frame is? I need to find the contrasts and edges. Another thing we need to figure out is where objects are. So where is the bike? Is it to my right or is it in front of me? So my brain is going to need to be able to process real world space based on things like receptive fields and feature detectors. We next need to figure out, be able to differentiate different wavelengths of light so that we can process things in different colors, because colors are going to be really important to us as humans, and other animals also need to be able to differentiate red objects from green objects, etc. And then finally, what do I do with this information? I need to be then consciously aware of the information that I'm processing so that I can maybe recognize an object, right? Recognize something as a bike, right? Do complex processing with it, determine which way it's moving, how fast it's going, these kinds of complex processing of the objects in the world. So I need to solve all these problems. In this video, we're going to cover the first two, and then in the final video, we're going to cover the last two. So contrasts and edges are really important to artists who study how to visually present things to their clients or to the world. Here's some art by Kazimir Malevich, kind of a study on contrast and edges. You see we've got high contrast here, and the square stands out, and low contrast, and the square blends in. And if you've ever tried to draw something or outline something that you're coloring or put a frame around a picture, you know how important contrast is to differentiating the object from what surrounds it. Our brains need to have some mechanism for doing this, for differentiating that this is different from this, and even that this is different from this, even though it's a little bit harder. So we need some sort of biopsychology to do this. And what's really important to consider here is that differentiating one object from another is based on the contrast of what's in one part of our visual field and what's nearby in context. And our brain's going to be processing not only what it's looking at, but what it's looking at adjacent to. And contrast is going to be enhanced in our brains. This is a nice demonstration of that, where we've got one square here on your left side and one square on your right side. And one of these squares probably looks darker than the other one to you. Typically, this one looks darker than this one, right? And this is due to the contrast of the surround. So the lighter colored surround makes this appear darker, and the dark surround makes this appear lighter. But that's simply a matter of contrast. The squares are actually the same level of brightness. If you're not convinced, here's those two squares without the surround, and you can see they both look pretty dark. So we have a system for contrast enhancement, right? So when you've got dark surround and light on the inside, we enhance the difference between those so that we can see the differences between an object and what's around it. We actually amplify those differences and enhance the contrast in our brains. So simultaneous contrast is a nice example of that. There's another phenomenon called Mach bands that look a bit like this, where the intensity of the brightness in these bands are actually stepwise like this, where this is a little brighter than this is a little brighter than this, and the rectangles are the same all the way across. What people actually see are these little dips and spikes at the edges, right? Where there's a little light band here and a little dark band here at the edge, where this edge is getting enhanced in terms of the contrast. Here's a blown up picture of Mach bands in black and white to highlight that brightness contrast. And you can kind of see here where this number 4 band seems to be getting darker as it gets close to the edge over here. It seems to be getting lighter as it gets to the edge with number 3. It's not actually getting lighter over here, and it's not actually getting darker over there. Your brain is enhancing the contrast to make this look brighter and to make this look darker, so it's easier for you to see the difference between 3 and 4. So how does this work? Well, your retina, the cells in your retina are engaging in what's called lateral inhibition. Lateral means sort of like to the outside or to the sides, right? We actually discovered this with horseshoe crabs, who have a kind of similar visual system where light comes in and hits receptors, and then there's cells that cross them, just like our horizontal cells do. And what researchers found was if you take a recording from a single receptor, like you take a recording from receptor A, maybe this is a rod named rod A, right? And you shine light just onto A, what you find is that it'll fire an action potential. No problem, right? So if you show light into the single receptor, you get rapid firing. If I shine light not just to A, but also to these nearby neurons, receptors, what you actually see is that the firing of A goes down. It slows down. So it's not just more light is more activation. More light nearby actually reduces the activation. And if I turn the light up on B, it actually slows down even more. So what researchers proposed was that each of the cells, each of these rods, right, is being inhibited by activation of their neighbors. So if I shine a light on A, it's inhibiting B. If I shine a light on B, it's inhibiting A. So if I get really bright light that covers all the receptors, it's going to be relatively minimal firing of each individual one. But if I got focused light, right, then I'm going to get rapid firing just of that one. So you can see how this enhances contrast, right? When there's contrast and the light is over here but not over here, I'm going to get a big difference between A and B. But when there's no contrast, then I'm not going to get a difference. I'm going to get sort of slow firing overall. And so when you get an edge, like in the Mach bands, you're going to have either more or less inhibition because of the bright light coming from the bright sides, or the dark light coming from the dark sides. And then that's going to enhance those contrasts and edges. So that's why you put a dark frame around a light colored picture, right? Or a light frame around a dark colored picture, because it actually makes the picture stand out more physiologically, right? We're going to either inhibit or not inhibit the image to make it either darker or lighter, depending on the frame that's around it. Here's just another image of lateral inhibition going on at edges. You can see that the intense light is going to cause a lot of inhibition to the dim light. So when these cells get activated over here, they will inhibit these cells over here. Less so going this way because there's less light from this direction. And so you end up getting this pattern where it actually looks darker. It fires less on the dim side, and it looks brighter. It fires more on the bright side. So we've enhanced this edge because of the inhibition going in both directions. So we got this system, and it starts in your retina, where we can see edges and we actually enhance edges. The next step is to differentiate the object in space. Okay, so how am I going to map those objects in my brain? We have receptive fields for this. Now, a receptive field is the area of your visual field, the world that you can see with your eyes, right? Within which the appropriate visual stimuli influence the firing of a neuron. We have neurons in our brain, in our occipital lobe and in our thalamus. There are different parts that are going to be tuned to specific stimuli in specific locations of the world. Okay, so I've got neurons that will only fire to light in a certain area of the world at a certain angle. This was actually discovered with cats by researchers named Hubel and Wiesel. What they did was they were showing cats different objects, pictures of mice, circles, triangles, that kind of thing, to see which parts of their visual cortex responded best. And it turned out that the cat's brains didn't respond to objects like, differentiating a mouse from a triangle. What they responded to were like the edges of the slides that they were sliding down on the slide projector. So it was the edges, the very simple lines were getting different responses. So it seems like we have, and they were only getting responses in certain places. So it seems like we have individual neurons tuned to very simple things like lines in very simple places. We have these receptive fields that are attuned to space and direction throughout our retina geniculate striate system. So starting in the eyes to the thalamus, the geniculate nucleus of the thalamus, and then to the striate cortex of your occipital lobe. We've got similar receptive fields along the way. As I mentioned, they respond to light patterns in specific places. Importantly, they don't fire to bright light all the time. So some of them are actually going to respond when there's bright light in that area, and some will respond when the light is dark in that area as compared to outside. And this leads to on firing and off firing. Taking a look at that, we have some cells that do on-center firing, where if you have a light in the center of the visual field, so this is a particular place in the world, if there's light in here, then the cell will fire. If there's light outside of it, then it won't fire. Some cells actually work the opposite way, where they won't fire if there's light in the middle of it, but they will fire if there's light on the outside. This is actually really useful because some signals we're looking at are bright and some signals we're looking at are dark, and we need to be able to see them both. So for example, the letters on this page are actually dark in color, so they're not sending light from this area of my visual field, right? The light's coming from outside the letters. And so actually reading these dark colored letters requires me to have these like off-center cells, so that when I focus on the letter S, I'm actually getting firing because there's a surround that's bright and the inside is actually dark, right? So these off-center cells are going to fire when I'm looking at these dark letters. Same thing is going to happen when there's a black housefly flying around, right? Black fly flies around. It's dark in the center and it's brighter on the outside. And so when I look at the fly, it's actually going to be an off-center cell firing. So I don't just respond to light. I respond maybe sometimes to off-center light. As I mentioned, we don't just identify locations of light, but we're also going to detect features, including certain angles. I'll show you a brief video here of what Hubel and Wiesel discovered with cats. What you're going to hear here is a bunch of clicking noises. the clicking are action potentials, and those action potentials that you hear right there are going to fire when this light crosses a certain space at a certain angle. What you're going to notice is that the action potential, this specific feature detector in the occipital lobe of the cat, is only going to fire when the light is at that angle and in that place. It's not going to fire when the light goes just above it or just below it. It's very specific. And in fact, when I put light outside it and even larger, I can define the barriers. Okay? Where is this receptive field end? You can see him mapping this on out. Okay, so this is what's known as a simple cortical cell. It's an individual neuron in the occipital lobe that's going to respond only to light at this angle in this place. You can see on the left here a few examples of simple cortical cells that are, again, on or off versions of them, right? They're going to be at a particular angle, and only a certain space in the visual field is going to fire. Your book talks about another kind of cell that also shows up in your occipital lobe. They're actually more common, called complex cells. They're really only complex in that they're going to cover, they're going to be less space dependent, right? So those complex cells are going to fire when I've got this bar here, or here, or here, right? So a simple cell has to be a bar of a specific kind in a specific place, whereas complex cells respond to a feature, like a line at this angle, but it can be anywhere in your visual field. This is the kind of complexity that we're talking about here. Check out your lecture slides, and your book has this resource as well for another video kind of breaking this down, how the features detectors work, simple and complex cells, and then how other features they can detect, like color and that sort of thing. But otherwise, that's going to get us a basics of what we're looking at. In the next video, we'll actually get more complexity in terms of how we see actual things that we recognize and how we see complex movement.